Okay, we're back. We were talking about the endocrine pancreas. We had talked a lot about insulin. Remember that insulin is the hormone that is used in the postprandial or post-absorptive state, the absorptive state, where you have a ton of nutrients flowing through your blood and you need to get them put away, particularly glucose. But we're now going to switch over to the other side of the endocrine pancreas and talk about glucagon, and we're also going to talk about other hormones that act against insulin. So this slide reviews what insulin was all about. Your high blood sugar that's caused by eating increases insulin, which comes from the beta cells of the pancreas, which acts on the liver, muscle, and fat cells to convert sugar to fat and sugar to glycogen. Fat mainly in the adipose tissue, but also somewhat in the liver, and glycogen mainly in the liver and the skeletal muscle. So insulin puts nutrients away, particularly glucose. Okay, in opposition to insulin, when you haven't just eaten, when it's been a while since you've eaten, then we have what we call the moving the screen a little bit. Then we have a situation where you're starting to run out of blood glucose, where your muscles and your brain and your other tissues need some glucose, and we have to free that up from the stores that are in the body. So besides insulin, all the other hormones that are involved in energy metabolism are in opposition, and these are collectively called glucose counterregulatory controls, or the short version is anti-insulin hormones. Of these, glucagon is definitely the most important. It's the most significant. But we also have epinephrine, glucocorticoids, thyroid hormones, and growth hormones acting in opposition to insulin. So there's 5 different hormones working against insulin. The first of these, and the most important, especially since we're talking about the endocrine pancreas, and this one comes from the pancreas, is glucagon. Glucagon is a protein, a peptide hormone. Its final form is 29 amino acids, but it actually has more than one form, around 3.5 kilodaltons, produced by the alpha cells of the pancreas, where insulin was from the beta cells. Synthesized as a pre-pro hormone, just like many other hormones, goes in, the messenger RNA attaches to ribosomes on the rough endoplasmic reticulum. The nascent peptide, the pre-pro hormone, is secreted as it's synthesized into the lumen of the ER. This gets packaged into vesicles that travel to the Golgi, which gets repackaged into vesicles and travels to wait alongside the plasma membrane for a signal for release, which is always going to be calcium. There's several different glucagons. There's not just one. Very highly conserved among vertebrates. It's an important hormone. Low sugar is going to increase glucagon. Remember, high sugar was what was going to cause insulin release. Low sugar is going to cause glucagon release, causing the liver to convert glycogen to glucose. It's one of the major pathways, but it has a number of different actions. So remember that the insulin put a whole bunch of glucose into glycogen. Now glucagon is going to convert glycogen to glucose. So the scenario is that low blood sugar promotes glucagon release from the alpha cells of the pancreas. Glucagon acts on the liver to stimulate glycogen breakdown. Glycogen is converted to glucose, and this goes into the blood, and this gives you higher blood sugar. Glucagon is released from the alpha cells under low glucose through a mechanism that isn't 100% understood. There's some parts of it that we don't know. We do know that low glucose is going to be the trigger, and we do know that insulin and somatostatin are anti-glucagon hormones. Somatostatin is anti-everything. It's just an inhibitory hormone everywhere where it's released. But insulin is also going to, it just makes sense that insulin will inhibit glucagon. Glucagon's released from the alpha cells. I don't want to go through all this pathway, but if you look at the left side of this figure, low glucose, which is coming in through the GLUT1 transporter, is going through a mechanism that's not completely known to cause a depolarization that results in increased calcium. That's the bottom line. So you're going to have some changes in membrane potential through a receptor that isn't really quite fully understood, or a mechanism that isn't understood. That's going to cause calcium to increase inside the cell, and that's going to cause exocytosis of the glucagon-containing vesicles, and you're going to get glucagon release. Under high glucose, this mechanism doesn't operate. So low glucose, you have a ATP to ADP ratio that's going to be low because the glucose isn't making any ATP, so you're going to have a lot more ADP. That seems to inhibit facilitate glucagon secretion. High ATP is going to inhibit glucagon secretion. The activity of glucagon in the liver is going to be somewhat predictable. We're trying to raise blood sugar. So we're going to activate these glucagon receptors by glucagon binding to it. It's a G protein coupled receptor, and it's coupled to G-alpha S, which is, as you know, stimulates adenylate cyclase. Adenylate cyclase is going to increase cyclic AMP. Cyclic AMP is going to increase protein kinase A. Protein kinase A is going to turn on other kinases, and ultimately we're going to get phosphorylation of glycogen synthase, which inactivates glycogen synthase. So you're not going to make any more glycogen. You don't want to make glycogen when you're trying to break it down. You're going to get induction of gluconeogenesis, which is production of new glucose from scratch. And you're going to get glycogenolysis, which means breakdown of glycogen into glucose. That's mainly through, as we'll see, other enzymes that are turned on. Glucagon, this is kind of redundant. Like I said, I like to put some redundancy into my slides. Glucagon acts through a G protein coupled membrane receptor that activates adenylate cyclase, increases cyclic AMP. Cyclic AMP activates protein kinase A. This results in a phosphorylation cascade turning on a bunch of proteins. One of the proteins that's activated is phosphorylase A. Phosphorylase A converts glycogen to glucose 1-phosphate. Glucose 1-phosphate is easily converted to glucose. So the glucose then moves out of the cell through transporters and into the blood. The effects of glucagon oppose those of insulin. So in addition to this glucagon is glycogenolytic, so it breaks down glycogen through the phosphorylase A that we just talked about. It's gluconeogenic, which means it promotes synthesis of new glucose from scratch. Okay, it makes new glucose. And it's lipolytic, means it breaks down fats. Now, that action is not going to be as much in the liver, although the liver does store some fat. It's also going to be in the adipose tissue. Glucagon causes glycogen breakdown in liver, but not in muscle. Your glycogen in muscle is saved for fight or flight, and it's going to take epinephrine to get the glycogen out of muscle. Glucagon frees up glucose from breaking down glycogen in the liver, which stores quite a bit of glycogen, by the way. Glucagon acts on adipose tissue, your fat tissue, to increase fat breakdown, and in turn that results in increased ketogenesis by the liver. The liver produces a lot of ketones, and those ketones can be used for energy by a lot of cells. Nervous system likes to use glucose itself, but other cells can use all kinds of energy sources. Glucagon increases gluconeogenesis, as we talked about before, glucose from scratch. Now, glucagon works through a G-alpha stimulatory. Epinephrine, which works along with glucagon under similar situations, mainly fight or flight situations, also works through G-alpha S in liver and muscle. Glucagon mainly acting on the liver, epinephrine mainly acting on the muscle, but they both work through a G protein coupled receptor. That's going to activate the G-alpha S. G-alpha S is going to turn on adenylate cyclase. Adenylate cyclase is going to activate protein kinase A, or make cyclic AMP. That activates protein kinase A, and that phosphorylates other proteins. If you follow the chain down, you'll see that one of the proteins that gets phosphorylated is glycogen synthase. So you don't make any new glycogen, and also in the phosphorylase A, which is going to make convert glycogen into glucose 1-phosphate, and the glucose 1-phosphate is going to be made into glucose. This is a redundant figure from the previous one, but it just shows you that in hepatocyte, there's receptors for epinephrine that are coupled to adenylate cyclase. There's receptors for glucagon that are coupled to adenylate cyclase. Both of them are going to activate adenylate cyclase, convert ATP to cyclic AMP. Protein kinase A is going to be activated. That's going to activate phosphorylase B kinase. which is going to activate phosphorylase A, which is going to convert glycogen to glucose 1-phosphate, and that'll be glucose that goes into the blood. Glucagon and epinephrine are synergistic, so they really increase this in the liver. But glucagon doesn't work on skeletal muscle, but epinephrine does. So the same process occurs in skeletal muscle, but only with epinephrine. So we get glycogenolysis and gluconeogenesis. A lot of redundancy here, but these are things that I wanted you all to know. I gave you a sheet before with a lot of definitions on it. So glycogenolysis is breakdown of glycogen into glucose, occurs in the liver under glucagon control and in the muscle under epinephrine control. Liver converts glycogen into glucose 1-phosphate. Muscle converts glycogen into lactate, lactic acid. That travels to the liver and gets converted to glucose in the liver. So it's a two-step process for muscle. Gluconeogenesis is new synthesis of new glucose from scratch, and this only occurs in the liver under glucagon control, but it also occurs in the kidneys, probably not under glucagon control, but as a regular part of your normal physiology. It's thought that in type 1 diabetes, this is a real problem with kidneys producing a ton of glucose from scratch. Maybe 50% of your blood glucose may come from your kidneys in type 1 diabetes when you really don't need any more glucose. The net effect of glucagon activity is increase in plasma glucose, increase in fatty acids, which can be used for energy by the cells, and increase in ketones, which can be used for energy by the cells. Epinephrine is another anti-insulin hormone. We've already talked about it to some. And glucose counter-regulatory hormone comes largely from the adrenal medulla, but also from sympathetic innervation to the liver and adipose tissue. Sympathetic nerve endings produce about 80% norepinephrine and about 20% epinephrine. So you get that. Epinephrine opposes insulin action by promoting glucagon secretion. So epinephrine helps make more glucagon. Epinephrine increases glycogenolysis in both liver and muscle, synergistic with glucagon in liver and by acting by itself in muscle. Increases gluconeogenesis in the liver, increases lipolysis in the adipose tissue, so fat is freed up. This is a very important effect of epinephrine. Decreases glucose uptake by muscle. You're trying to free up glucose, not use it up. And it stimulates adrenocorticotropic hormone secretion, ACTH, resulting in increased glucocorticoids. So epinephrine is making glucocorticoids, which are also an anti-insulin hormone. The glucocorticoids' role in energy metabolism or increased protein catabolism in peripheral tissues. So you're breaking down protein. That results in increased amino acid levels in the blood. These can be used for energy. They can be uptaken by the liver. Stimulation of gluconeogenesis by the liver. This may be permissive, allowing glucagon and epinephrine to work, particularly epinephrine. Inhibition of glucose uptake and use by peripheral tissues. You don't want to be taking up glucose while you're trying to use it. And increased glycogen synthesis by the liver, which is kind of counter to what we would think would be happening at that time, but that's one of the effects of it. The main effects of thyroid hormone are increased absorption of glucose from the intestines. Thyroid hormone is really important for getting glucose absorption increased. Depletion of liver glycogen. This can be very life-threatening in hyperthyroid situations. So if you have Graves' disease or similar hyperthyroid situation, one of the life-threatening aspects of it, if it's not treated, if you don't shut the thyroid down, is to completely deplete your liver glycogen, causing liver disease. Thyroid hormones accelerate degradation of insulin, so they shorten the half-life of insulin. Okay, so that's pretty much everything for the endocrine pancreas. Stay tuned for note set 15, which is type 1 and type 2 diabetes. It's also a short one, so I'm trying to make these a little bit shorter so that they tell me that the average undergraduate has an attention span of 15 minutes. I think it's probably more like 5, but that's just my opinion. So by shortening these, I'm hoping that you can stay with me a little bit longer. All right, take care.